Let's talking about mycology today. The term mycology is derived from Greek word mykes meaning mushroom. Therefore mycology is the study of fungi. Mycology is the branch of biology concerned with the study of fungi, including their genetic and biochemical properties, their taxonomy and their cause humans illness, such as toxicity or infection. The ability of fungi to invade plant and animal tissue was observed in early 19th century. But the first documented animal infection by any fungus was made by Agostino Bassi. Agostino Bassi, sometimes called Delodi, September 25, 1773 to February 8, 1856, was an Italian entomologist. He preceded Louis Pasteur in the discovery that microorganisms can be the cause of disease. He discovered that the muscardine disease of silkworms was caused by a living, very small, parasitic organism, a fungus that would be named eventually Beauveria bassiana in his honor. In 1844, he stated the idea that not only animal, insect, but also human diseases are caused by other living microorganisms. For example, measles, syphilis, and the plague. Historically, mycology was a branch of botany. Because, although fungi are evolutionarily more closely related to animals than to plants, this was not recognized until a few decades ago. Many fungi produce toxins, antibiotics, and other secondary metabolites. Fungi and other organisms traditionally recognized as fungi, such as oomycetes and myxomycetes, slime molds, often are economically and socially important, as some cause diseases of animals, such as histoplasmosis, as well as plants, such as Dutch elm disease and rice blast. Some fungi can cause disease in humans and other animals. There are two types of fungi, yeasts and molds. Yeasts grow as single cells that reproduce by asexual budding. Molds grow as long filaments, hyphae, and form a mat, mycelium. All fungi have typical eukaryotic morphology. They have rigid cell wall composed of chitin. Inner to the cell wall is the plasma membrane that is a typical bilayered membrane. The cytoplasm consists of various organelles such as mitochondria, Golgi apparatus, ribosomes, endoplasmic reticulum, lysosomes, microtubules, and a membrane-enclosed nucleus. A unique property of nuclear membrane is that it persists throughout the metaphase of mitosis, unlike in plant and animal cells where it dissolves and reforms. The nucleus possesses paired chromosomes. Several medically important fungi are thermally dimorphic, such as they form different structures at different temperatures. They exist as molds in the environment at ambient temperature and as yeasts, or other structures, in human tissues at body temperature. Most fungi are obligate aerobes, some are facultative anaerobes, but none are obligate anaerobes. All fungi require a preformed organic source of carbon, hence their frequent association with decaying matter. The natural habitat of most fungi is, therefore, the environment. An important exception is Candida albicans, which is part of the normal human flora. Asexual reproduction is the most common pattern among most fungi. And fungi only participate in sexual patterns in certain situations. Pathogenesis of fungal diseases, mycoses. Most fungi are saprophytic or parasitic to plants and are adapted to their natural environment. Infection in humans is a chance event, occurring only when conditions are favorable. Infection depends on inoculum size and the general immunity of the host. Host defense factors including physical barriers, such as skin and mucous membranes, the fatty acid content of the skin, the pH of the skin, mucosal surfaces and body fluids, epithelial cell turnover, normal flora, chemical barriers, such as secretions, serum factors. Most fungi are mesophilic and cannot grow at 37 OC. Natural effector cells, polymorphonuclear leukocytes, and the professional phagocytes, monocytes and macrophages. Factors predisposing to fungal infections. Prolonged antibiotic therapy. Underlying disease, HIV infection, cancer, diabetes, etc. H. Surgical procedures. Immunosuppressive drugs. Irradiation therapy. Indwelling catheters. Obesity. Drug addiction. Transplants. Occupation. Immunity to fungal infections. Mechanism of immunity to fungal infections can be innate or acquired. The nonspecific immunity includes the physical barriers offered by skin and mucous membranes along with their secretions and normal flora. Cell-mediated immunity. Immunity is provided nonspecifically by effector cells, polymorphonuclear leukocytes, and professional phagocytes, monocytes and macrophages, and specifically by T lymphocytes. The phagocytes are very important in defense against Candida, Aspergillus and zygomycetes. Humoral immunity. Even though antibodies are produced against many fungi, their role in protection is not very clear. However, antibodies help in clearing fungal pathogens through opsonization, which is important against Candida and Cryptococcus. Another component of humoral immunity is the complement, which can act as opsonins and may even cause damage to their cells through complement activation. Antibodies are important to fungal serodiagnosis. Pathogenesis. The response to infection with many fungi is the formation of granulomas. Granulomas are produced in the major systemic fungal diseases. The cell-mediated immune response is involved in granuloma formation. Acute suppuration, characterized by the presence of neutrophils in the exudate, also occurs in certain fungal diseases. Fungi do not have endotoxin in their cell walls and do not produce bacterial-type exotoxins. Intact skin is an effective host defense against certain fungi, such as Candida, dermatophytes. The normal flora of the skin and mucous membranes suppress fungi. When the normal flora is inhibited, such as by antibiotics, overgrowth of fungi such as Candida albicans can occur. Fungal toxins and allergies. In addition to mycotic infections, there are two other kinds of fungal disease. 1. Mycotoxicoses, caused by ingested toxins. And 2. Allergies to fungal spores. The best known mycotoxicosis occurs after eating Amanita mushrooms. These fungi produce 5 toxins that cause pronounced vascular and neurologic effects. Other ingested toxins, aflatoxins, cause liver damage and tumors in animals and are suspected of causing hepatic carcinoma in humans. Aflatoxins are ingested with spoiled grains and peanuts. It leads to a loss of p53 protein and a consequent loss of growth control in the hepatocyte. Allergies to fungal spores, particularly those of Aspergillus, are manifested primarily by an asthmatic reaction. Laboratory diagnosis. There are 4 approaches to the laboratory diagnosis of fungal diseases. 1. Direct microscopic examination. 2. Culture of the organism. 3. DNA probe tests. And 4. Serologic tests. Direct microscopic examination of clinical specimens, such as sputum, lung biopsy material, and skin scrapings depends on finding characteristic asexual spores, hyphae, or yeasts in the light microscope. Antifungal therapy. The drugs used to treat bacterial diseases have no effect on fungal diseases. The most effective antifungal drugs, amphotericin B and the various azoles, exploit the presence of ergosterol in fungal cell membranes that is not found in bacterial or human cell membranes. Amphotericin B, fungizone, disrupts fungal cell membranes at the site of ergosterol, and azole drugs inhibit the synthesis of ergosterol, which is an essential component of fungal membranes. Another antifungal drug, caspofungin, CANCitis, inhibits the synthesis of beta-glucan, which is found in fungal cell walls but not in bacterial cell walls. Human cells do not have a cell wall. This is the basic mycology. Thanks for watching. If you like this video, please thumb up and leave a message. You must be my biggest motivation to make the video. Please subscribe to the channel, where you'll find more and more medical videos here later. Thanks.